In the next 50 years, global population is expected to double, reaching more than 8.9 billion people. Population growth and improved diets will require at least doubling of the food supply. In connection to the emerging challenges posed by the rapid growing human population, a need for the implementation and the use of advances in science and technology is inevitable. Livestock biotechnology provides a solution to the increasing demands for quality and safe milk, meat, and other animal byproducts. Recent breakthroughs in biotechnology are the DNA-based or marker-assisted technologies, which are applied in various areas of livestock production, such as
animal breeding aims at improving the genes of animals to make them more useful to us. The main objectives of animal breeding are improving the growth rate of animals, increasing the production of milk, meat, egg, wool, and other products from animals, improving the quality of products, for example, improving the quality of milk or wool we get from animals. Improving the resistance to diseases. Animals will live longer and yield more products during their lifetime. Increasing the span of productivity. Increasing the rate of reproduction. For example, cow breeds are ensured to have successful and multiple reproductions in their lifespan. There are 3 approaches for animal breeding: inbreeding, outbreeding, and hybridization. Let me explain what are they. Inbreeding is the crossing of the male and female individuals of the same species among the same breed. Superior males and superior females in the breeds are identified for this purpose. Outcrossing involves crossing of superior males of one breed with superior female of another breed. It allows the desirable qualities of the 2 breeds to appear in offspring. In hybridization, superior males and females of 2 different species are mated. The progeny is often different from both the parental species. Apart from conventional breeding techniques where superior male and female are selected to obtain superior quality of offspring, genetical modification is also adopted for breeding. The conventional selective breeding technique in animals take long time to yield the desired results. Just as in plants, certain biotechnological procedures have been extended to animals also. The recombinant DNA technology has been used in animals also to introduce new genes into animals. Let me explain you the cases where animals are genetically modified to receive improved quality in products we get from animals. Certain human gene sequence introduced into cattle, goat, and sheep to get enhanced growth, meat, and milk production. Human gene in cattle is responsible for production of growth hormones in cattle. These hormones shows up in milk they produce. Milk will contain human growth hormones. Bacterial genes introduced into sheep to obtain improved wool output.
The use of these biotechnologies by animal breeders is not currently the focus of much public controversy. However, the prospect of cloning and genetically engineering animals has created a great deal of public discourse and debate. Often these 2 terms are mistakenly lumped together as being the same. However, these 2 procedures differ. Genetic engineering is the introduction of DNA sequences into the genome of a living organism by cutting a fragment of DNA from one organism and pasting it into the genome of another. Because the genetic code, or DNA, for all organisms is made up of the same 4 nucleotide building blocks, this means that a gene makes the same protein whether it is made in an animal, a plant, or a microbe. Genetically engineered animals are sometimes called transgenic, and they are able to produce the protein that is encoded by the inserted DNA sequence. Microinjection of foreign DNA into newly fertilized eggs has been the predominant method used for the generation of transgenic livestock over the past 20 years. To be passed on to the next generation, the transgenic or inserted DNA must be present in the organism's germ cells, that is, the eggs or sperm.
The first stage in the genetic modification of animals is the collection of fertilized egg cells at the stage where they contain two nuclei, before the fusion of the sperm nucleus with the egg cell nucleus. The new DNA can be introduced into fertilized egg cells by a variety of methods. One of them, known as microinjection, involves inserting the DNA directly into the male pronucleus. In another method, the new gene is placed in a retrovirus. This is then used to infect the egg cells. Although more effective, this method only enables the incorporation of short DNA fragments. The fertilized egg cells that contain the desired gene are then implanted into a female organism. The offspring are then selected by checking for the presence of the DNA of the specific gene in their cells. The polymerase chain reaction, PCR, method is usually employed for this purpose.
Cloning, on the other hand, is almost the opposite. It's the reproduction of an animal that is a genetic copy of another animal. Cloning is the process by which a genetically identical plant or animal is produced asexually. Sexual reproduction involves of an egg and a sperm, and asexual reproduction is in the absence of an egg and a sperm. Many of us have cloned without realizing that we were doing so. If we've taken a cutting from a plant and stuck it in the ground to root, we've cloned. We've produced a genetically identical individual, a plant that's identical to the one from which the cutting came, and we've done so asexually.
In animals, only embryo stem cells have the capability to differentiate all the different types of cells found in an adult. There are two methods you can use. The first is called embryo cloning. A farmer might have a cow that gives a lot of milk and wants to use her to create many calves. The cow is artificially inseminated using the sperm of a bull. The embryos grow until they form a ball of embryonic stem cells. Before they become specialized, the embryos are removed from the uterus, divided up into separate embryos, which will all have the same DNA, and then each is placed into the womb of a different cow. These cows are surrogate cows. They are just being used to grow the embryos until they are born. The calves will be clones of each other, but not a clone of the mother due to the bull's sperm. Hopefully, the calves will have the characteristics that the farmer desires, but as sexual reproduction is involved and the genes from both parents are randomly mixed, there is always the chance that they won't. There is a way to get around that problem, and that is to use another technique called adult cell cloning. This is used to form a clone of an adult animal. You might have heard of Dolly the sheep. She was the first mammal to be cloned back in 1996. There are several steps to this process. An unfertilized egg cell is taken from an adult female, and the nucleus is removed. A body cell, such as a skin cell, is taken from a different adult. The nucleus is removed from this adult body cell and is inserted into the egg cell. An electric shock stimulates the egg cell to divide to form an embryo. These embryo cells contain the same genetic information as the adult body cell. When the embryo has developed into a ball of cells, it is inserted into the womb of an adult female surrogate to continue its development, so 3 different adults are used. The baby born will be a clone of the adult that donated the body cell because it has the same DNA.
Meanwhile, wide arrays of modern reproduction technologies, including in vitro fertilization and embryo production, oocyte and embryo cryopreservation, intracytoplasmic sperm injection, ultrasound-guided ovum pickup, superovulation, and embryo transfer, are known to be valuable in livestock production for maximizing the potential of outstanding females for reproduction.
Another use of biotechnology in animal agriculture is embryo transfer. Through the use of reproductive hormones, cows can be induced to produce a large number of eggs, which can be recovered and, once fertilized, transferred to a number of different surrogate females. This allows for greater utilization of superior genetics from individual cows.
The next step of IVF is to perform an embryo transfer. This procedure is done in a transfer room near the laboratory.
And we actually describe it, it's quite similar to a pap smear and even less traumatic than a pap smear, because when we transfer the embryo, we don't want there to be much uterine cramping. So there's a speculum just like a pap smear, and then the embryo is just placed very gently through the cervix and then towards the top of the uterus. We do that under ultrasound guidance, which is nice for both us to assure that it's in the right place and also for patients to see the flash of the embryo coming out of the catheter. It's quite gratifying to see that.
In the past, when IVF was less efficient, more embryos had to be transferred to get a decent pregnancy rate. But with advancements in IVF, including embryo culture techniques and selection of embryos, we've shown that we can really achieve very high pregnancy rates in many cases by transferring a single embryo and have a better outcome for the patient.
On the other hand, semen cryopreservation and artificial insemination are technologies that capitalize on superior males for genetic improvement in livestock.
Artificial insemination is a biotechnology that has been extensively applied to improve the genetics of farm animals. Artificial insemination refers to the process of getting a female pregnant by physically placing sperm into her reproductive tract rather than by natural mating. Modern techniques for artificial insemination were first developed for the dairy cattle industry in the 1940s. This enabled the impregnation of many cows with the sperm from a single bull, maybe one with genes for high milk production. One of the main benefits of artificial insemination is that a single semen collection, which would normally be used to breed only one female, can be divided and used to breed multiple females.
A very important biotechnology for animal breeding is artificial insemination. Artificial insemination allows superior animals within a population to have many more offspring. Therefore, the genetics of a population can change more quickly. The problem of using this technology, or at least the constraint, is that there needs to be better data to select the animals to be used in artificial insemination programs. Yes, with artificial insemination, each male animal can produce more offspring. Therefore, you can choose the male animals that have the traits which will allow a given population to adapt more quickly to climate change. And when this animal has more offspring in the population, then in the next generation will be better adapted to climate change. Well, actually, this technology is available almost across the world. The one constraint I see, though, in the developing countries is that to apply this technology in an optimal manner, you really need to have good information about your populations. And that is the aspect that is really lacking in developing countries. You need to have more accurate information about the productivity, particularly of the local breeds, so that you can choose the most superior males within the population.